All right, we are now to topic 9. That means we just have a short review for topic 10. Topic 10 is just going to be linked genes and a little bit about mitochondrial DNA, and I'll do a chi-square practice for you just in case. So what I want to do is just go over chapter 11, Mendel and the gene idea, and then I'm going to put X-linked traits in there at the very end, and then I'll do a practice FRQ. All right, so you have a handout that goes with this. College Board is very unlikely going to ask you anything about Mendel, but I still think to give him respect. We're still going to talk about him, but if you don't want to write anything down for this question, it really won't matter. You're not going to get a question about Gregor Mendel. That would be something that an on-level high school biology class would be, not AP Bio. All right, so he studied peas. Remember, the reason he studied peas is because he could control the mating, and there's many varieties available in short generation time. It's very important that you know what P generation is, F1 and F2. P generation are going to be your pure breeding parents, meaning that they're homozygous for that trait. So with purple flowers, they would be homozygous dominant, and with white flowers, they would be homozygous recessive. Your F1 generation is going to be your heterozygous hybrid. All of the offspring are going to be heterozygous dominant allele with a recessive allele, and they're going to, in this situation, going to show the dominant trait. The F2 generation is the offspring from mating two F1s together. All right, so if we were doing this Punnett square, we would have, and I'm sorry, I don't have the pencil thing up, whatever. Just visualize it in your head. Big P, little p, crossed with big P, little p. And if you want to make a little Punnett square with that, so big P, little p on the top, big P, little p on the side. And this is 7th grade Punnett square, so you don't really even need to do it. So you would get 25% big P, 50% heterozygous big P, little p, and then 25% little p. And that would result in 75% purple and 25% white. So I would just definitely know the difference between P, F1, and F2. And there you go. This is posted on Veracross, just in case you're wondering. All of these are always posted. All right, definitely the definition of an allele, alternate versions of a gene, and it's important to remember, and you saw with the FRQ that we did last week, you need to know that any individual is going to have 2 alleles for every gene, one from mom, one from dad. So that's why with the mosquitoes, if you test for an allele in 39 mosquitoes, well, how many total alleles do 39 mosquitoes have? 78. So that's why we used that number 78 last week. All right, these are the characteristics that Mendel studied. Pretty easy, if you'll remember. Okay, and he has all these principles. There's alternate versions of genes that are alleles. You get 1 allele from each parent. If you have 2 different alleles, one's dominant, one's recessive. And then the law of segregation is the 2 alleles for each character separate during gamete formation. So that goes back to meiosis. All right, I'm just going to kind of skip a couple of slides here. I think homozygous, heterozygous, you guys know that. Hopefully, you do. I know you do. Phenotype is, I would just definitely brush up on these 2 words. Phenotype is the expressed physical trait. So you think phenotype is the physical trait, it's what you can see. Genotype is the actual genetic makeup. So phenotype is a description. Genotype are just the letters, all right? And here's an example. Phenotype, purple and white. Genotype, you have your letters. Okay, and then we've done Punnett squares. A test cross, I think it's important to know what the word test cross means. It means if you have something that is showing the dominant trait, but you don't know, is it homozygous dominant or is it heterozygous? Well, let's test by crossing it with a known homozygous recessive. If you get any recessive phenotypes, that means that that original one that you're questioning had to be heterozygous, right? Because if it's carrying a recessive allele, it's heterozygous. So with things like recessive traits in humans, So dark colored eyes would be dominant, light colored eyes would be recessive. Now there's a lot that goes into inheritance of eye color, but you can, with blues and greens and all of that stuff and hazel, but you can pretty much say light is recessive, dark is dominant. Let's say you have a dark-eyed woman and a light-eyed man, and you're like, I wonder, are the kids going to have light eyes or dark eyes? If those kids come out with light eyes, you can pretty much guarantee that the woman who had brown eyes was heterozygous somewhere for the light eye colored gene. Anyways, I'm just trying to relate that to your lives as much as possible, even though you're not worried about that right now, probably. Anyway, law of independent assortment. Each of the alleles separates independently. This goes back to meiosis, where chromosomes line up on the metaphase plate independent of each other, and they segregate independently. I just don't think you'll be asked about this law of independent assortment. Man, I hope not. It's so hard to explain. So the way it's explained in this picture is that you have this beginning germ cell right here. Remember, germ cells are diploid cells that are in the sperm, not in sperm, sorry, that are in the ovaries and the testes. And you see the blue ones and you have the red ones. Well, here's one scenario of how they could line up on the metaphase plate. And then here's the ultimate outcome of the gametes. Well, here's another just as likely outcome. They can line up a different way, and you can see you end up with completely different gametes. So, all right, a monohybrid cross is one character, a dihybrid cross is two characters. And I think we're getting to the next question here. Sorry, my chair is making noise. How would you solve a dihybrid cross that have both heterozygous parents? A dihybrid cross with both heterozygous parents, here's your example right here. I hope everyone can remember if they are double heterozygous for two different traits, that means that you can use the 9331 rule. Okay, so I would do that. Sorry, I just got an email and I was reading my email while I was talking. Yeah, nice. So 9331, that's how you would do that. So that's how you need to answer that question. All right. How would you solve a dihybrid cross that had big A, big B, little a, big B, little b? What you would do is you would reduce them down. So for that first one, your gametes could only be big A, little b, and little a, little b. So you would just do a cross with those two gametes. And then for the other parent, the only gametes possible would be little a, big B, and little a, little b. So that's how you would do that cross. So pretty easy. If you don't remember how to do those, let me know. Unfortunately, I didn't take a picture of the work. All right, let's see. Rule of multiplication and rule of addition. So if you have the word and, so things occurring together, you have the rule of multiplication. I have a feeling that's not going to show up on there, though. I know I'm giving you all these things that aren't going to show up. I'm just going through the original slideshow, trying to eliminate as much as I can. And it's helpful to remember the rule of addition. It would be when you have different situations where the word or is used. I just don't think they're going to put that on there. But anyways, I'm just going through it. I'm not going to go over a coin toss. So complete dominance is the example of the purple flower, white flower, the purple flower being completely dominant, meaning if there's just one dominant allele, it will have a complete dominant phenotype. So it's not just going to be like a light purple, it's going to be a dark purple, whether or not it's homozygous dominant or it's heterozygous. Okay, I'm looking at the handout that I gave you, make sure I don't skip over anything. Okay, we're on give an example of complete dominance. Okay, so you have give an example of codominance, and then I'll have to go. Oh, give an example of incomplete dominance. Sorry, I'm a little bit disorganized. Incomplete dominance would be the other example where you would see a dark flower, a dark purple flower, and a white flower come together to form a hybrid that would be like a light purple. That would be incomplete. It's an in-between. Or if I can use examples of chickens, a black chicken and a white chicken come together to create a hybrid that's a gray chicken. All right, that's incomplete. It's an in-between. Codominance is where you see both alleles. So the Rowan horses Remember from far away, a roan horse is going to look reddish, pinkish actually. But what it is if you plucked out a bunch of hairs from these horses or cows, they can show this trait too, this hair color trait. And you actually looked at the hairs, you could divide them up into white and red hairs. It appears to be incomplete dominance, but it's actually codominance. You have completely white hairs and complete red hairs. When if this was incomplete dominance, every single hair would be like a pink color. Hopefully that makes sense. Another example of codominance would be AB blood types. If you have the allele for A and you have the allele for B, you have AB blood. And it shows you right here what blood typing even means. They're carbohydrate chains that are hanging off of the red blood cell. And so if you have type A, you have a certain type of carbohydrate. If you have type B, you have a certain type of carbohydrate. And if you have AB, you just have both, and both of them are being expressed. So AB blood type is an example of codominance. ABO blood type is an example of multiple alleles. So how many different alleles are available for blood type? Well, it would be 3. You have type A, or you have allele A, you have allele B, and then you have this allele I. And so what the allele I means is no carbohydrate chains hanging off of the red blood cell. I don't think you'll get, I think just understanding how you can get a certain blood type and understanding that it's codominance is what's best here. I just don't anticipate Punnett squares. That's usually something reserved for multiple choice questions on the AP Bio exam. All right. And then this tells you phenotype. Remember, phenotype gives you a description. Genotype tells you the actual genes, both alleles that are involved. All right, polygenic inheritance is something that's really important because, you know, when you're in 7th grade science and even when you're in cell biology, I'm pretty sure cell biology does not go into this idea. They might though. I guess they actually might, just depends on the teacher. I know I would. When I teach cell biology, I actually do go into this because I think it's so important. It's incomplete for students to think that one gene is responsible for one trait. It just doesn't work that way. There are multiple genes that can act upon a single trait. It's just not black and white or as cut and dry as people think. Oh, you have the gene for being tall or you have the gene for being short. No, there's multiple genes that go into how tall a person is or how short a person is. And obviously there's like a spectrum of how tall or short a person is in height. So skin color and height is polygenic inheritance, multiple genes acting on a single phenotype. And that's just your example of skin color, and they're talking about 3 genes that go into that. And then this just gives you some of those vocab words. What is epistasis? What is pleiotropy? So remember epistasis. I love this one because I like labs. I don't know, I had a lab before, but it didn't work out so well. I'm really bad with training dogs. My lab would jump on everybody. She was so crazy. So we got a basset hound, and you know, basset hounds have really short legs, so they can't jump on people. So no, I don't have to worry about training her for that. And basset hounds are really lazy, so like they just want to sleep all the time, so you don't have to worry about them being bad. Anyways, totally off topic, but I love doing the epistasis example because labs just usually make people happy, except for the one that I had. So if they have the dominant gene and for fur color and the dominant gene for the deposition of fur color into the fur, they will be black. Okay? So they have to be dominant for both in order for them to show the black fur. Now, if they have the recessive little b for fur color, but they're dominant for the deposition of the fur color, they will be brown. And then what's interesting is the yellow lab. If they are dominant for fur color or recessive for the fur color, black or brown, but they don't have the dominant gene for the deposition, you can see when you do these crosses right here, it's little e, It doesn't matter. It doesn't matter what color their genes code for. If they do not have the gene responsible for depositing that color into the fur, they're going to be yellow. That's epistasis. The phenotypic expression of one gene is actually affected by another. So it's this E. The E is actually going to affect whether or not the brown or the black gets expressed at all. I think that's really interesting. And then pleiotropy. Pleiotropy, remember, so if polygenic inheritance is multiple genes affect one trait, pleiotropy is one gene affects multiple traits. So it's kind of like the opposite. One gene. And then anytime, remember on the multiple choice, I was like, anytime it says a disease, if it's asking for pleiotropy, it's got to be the disease. Because even if like sickle cell is just one teeny tiny little base substitution that causes a change in an amino acid that causes red blood cells to be misshapen. And oh my gosh, that creates lots of different traits for that person. They, have to get amputations. They are tired all the time and stuff like that. So pleiotropy. Oh my gosh, someone asked on my AP Bio Facebook group, is there going to be pedigrees? How could they possibly do it? And another person said they have not put a pedigree on an FRQ in 8 years. Pedigrees do show up on the multiple choice, but they haven't shown up on the FRQs in 8 years. And gosh, I would hate to say that there's not going to be a pedigree, and then there is. The only thing is that how do you ask questions like, what's the control group? What's the independent variable? What's the dependent variable? And stuff like that. You just can't really ask that much on a pedigree. But just for completion's sake, circle is woman, square is man. If it's shaded, it means that they have the disorder. And what you do is you just kind of follow it along, and you remember me writing up on the Clevertouch, well, what could it possibly be? What mode of inheritance is this? So you have someone with a disorder, someone without the disorder. Half of their kids have it, half of their kids don't have it. Could that be a dominant disorder? Well, yeah, if she was heterozygous, dominant, that could be true. Could this be a recessive disorder? Well, yeah, if she was homozygous recessive and he was heterozygous, that still could work out. So what we look at here is we go kind of down the line. You have someone that has the disorder, someone that doesn't. Neither of their kids have it. And then they marry someone and have a kid and neither of them have it. Okay, cool. I just don't know if I can really conclude anything from this line right here. But look at this. If you look at this one, this gives you everything you need to know. She does not have the disorder. He does not have the disorder, meaning doesn't show it phenotypically. But poor little Carla, she has it. So that means it has to be a recessive disorder because it can be hidden. Because it's hidden in both parents, and then poor little Carla had it. Not going to go into this. I think I did enough. All right, last little topic is sex-linked genes. Thomas Hunt Morgan used the words wild type and mutant. What do they mean? Well, wild type means dominant, mutant means recessive. Wild type means dominant, mutant means recessive. So Thomas Hunt Morgan is the one that studied fruit flies, and he found that eye color, how could you forget that, lab? Eye color in fruit flies was X-linked. So red is dominant, white is recessive, and it's X-linked. And remember we saw if mostly males had the white eye because it's X-linked. And if a male, I feel like this word would show up if they talked about X-linked genes. They love doing X-linked genes on FRQs, just FYI. The word hemizygous is what is used for X-linked genes because a male cannot be homozygous. A male cannot be heterozygous for an X-linked gene because he only has one X. So he is considered hemizygous. When I see hemizygous, it makes me think like halfway. Okay, females can be affected or be carriers. Males can't be carriers because they only have one X chromosome. Right, here's an example of a Punnett square X-linked trait. So let's say mom is homozygous dominant for, we'll use color blindness. So she's not colorblind, she has normal color vision. Dad is hemizygous for color blindness. Do a cross, right? So here's dad up at the top. Here's mom over here. Cross them. And what do you get? Well, you get both daughters are carriers for color blindness now. Both sons, but both sons neither are carriers. They both got that big N, that dominant trait from mom. And so none of the offspring actually have color blindness in this situation. Now, let's go down here. Now, mom is a carrier, dad has normal. So mom's carrying color blindness, dad has normal. What is interesting to see if mom's a carrier is only one of her sons would be affected. Only one of her sons would be affected because he got that little N from her. All right, now let's say mom's a carrier and dad is showing color blindness. Now, dad has color blindness and mom's a carrier. Now you have a situation where you could have a colorblind daughter and you could have a colorblind son. This is, I don't want to say this actually isn't very common. It's not that common to see colorblind women. It's just not. And so I'm assuming this situation where she's a carrier and he's colorblind just doesn't show up that much. All right, and here's sex-linked disorder, color blindness, Duchenne muscular dystrophy, and hemophilia. You don't need to know any details about this, but you could see them show up as an example. All right, we have an FRQ. So what you can do right here is pause the video for this FRQ. Okay, so we're looking at this question. They're talking about fruit flies. Remember, fruit flies only have 4 chromosomes. They have sex chromosome, and then they have 3 autosomes, meaning it's a normal chromosome that doesn't determine sex traits. So what they're doing is 4 different genetic crosses for a particular X-linked recessive trait. When it says affected, that means it has the X-linked recessive trait and it shows it phenotypically. If it says unaffected, it means it does not show it phenotypically. As a female, the female could actually still be a carrier. 4 different crosses. And you see here an unaffected female with an affected male. Affected, unaffected, 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 affected, affected. Okay, cool. Percent of total population that are affected. And then you kind of notice a difference here. What looks like the total population is affected here more and here. Well, what do these 2 things have in common? The female is affected. Okay, well that makes sense. Okay. And then percent of males that are affected, it looks like 100% of males are affected if the female mother is affected in both circumstances. So if there's a disproportionate number of males that are affected from something that a mother has, that means it is an X-linked disorder. It already told you it was X-linked, but there's your verification. Percent of female offspring that are affected, only female offspring are affected if both parents are affected. Okay, and that makes sense, knowing it's an X-linked trait. Identify the genotypes of the male and female flies used in cross. Sorry, that should not say 22. That should say 2. Numbers get duplicated when I'm copying from the College Board website. So used in cross 2. Affected, unaffected, but males get the trait or get the disorder. So that would mean that the mother, okay, so mother is affected and sorry, female is, I don't want to call flies mom and dad. The female parent is affected and the female and the male parent is unaffected. However, 50% of the population is affected and 100% of the males. So if 100% of the males are affected, that means she is she has to be homozygous recessive. Even to be affected, she has to be homozygous recessive. So this would be X little r X little r for the mother. So she's not a carrier. She's completely unaffected. I'm sorry, she's completely affected. And for father or for male, the male parent, he would be X big R Y. Sorry, I totally butchered that question. All right, identify the cross in which the female parent was most likely heterozygous. So if the female parent is heterozygous for a recessive X-linked trait, it's either cross 1 or cross 3. So which one would she be heterozygous in, cross 1 or cross 3? Well, in cross 1, there's no males that are affected. So she can't, if no males are affected, she's not carrying the recessive trait. If she was carrying the recessive trait, there would be some males that were affected. So what we see here is in cross 3. And if you did a Punnett square where you had mom who was X big R, X little r, and then father who was X big R Y, you would have 50% of the males that were affected. Right, the researcher hypothesized that crossing any unaffected female and an affected male will result in a zero. I apologize again, my numbers get duplicated. Will result in a 0% chance of producing an affected male offspring. Evaluate the validity of the hypothesis. Right, so if we look back here, what they're saying, so they're saying an unaffected female and an affected male will produce 0% affected males. Unaffected female and an affected male. Okay, would always produce 0%. That's not true because if she was a carrier, um, she could produce an affected male. So that's what I would say is that the hypothesis isn't valid because if she was a carrier, so if she was X big R X little r, and then he is X little ry, you would end up with 50% of your males that are affected if she was a carrier. So that hypothesis is not valid. And you would give like a Punnett square as your answer for that. Explain how the results exclude the possibility that the trait is encoded by a mitochondrial gene. All right, so anytime you see anything about mitochondrial genes, you need to remember that mitochondrial genes are inherited from the mother. And so what that would mean if this gene came only from the mother, that it wouldn't matter if the father was affected or not. The offspring would have to have an identical genotype as the mother. So what you see here is an affected male and an unaffected. No, I don't want to look at that one. Well, they don't have one where there's an unaffected female and an affected male. But the answer to this one is if it's mitochondrial DNA, the mitochondrial genes are inherited from the female parent only, resulting in offspring that all have the same genotype as their mother. So they would have to all have the same genotype as the mother. And you see here that all the females are affected, yet there are some offspring that aren't affected. So that's why this can't be a mitochondrial DNA. All right, and that is it for this topic. Tomorrow I will cover um, linked genes, not X-linked, but linked genes, a practice FRQ for that. And I want to look at biotechnology really quick. I want to go over a couple of things with biotechnology because I forgot to throw that into the DNA review, and then a little chi-squared. So tomorrow is just more of kind of a wrap up of everything, and it will be the last review. All right, so thank you for watching this, and I will talk to you guys later.